Hello class and welcome to part 2 of our discussion for microbial nutrition, growth, and metabolism. So last meeting we have discussed the different elements found in living organisms. We also discussed some microbial growth factors which affects the growth as well as the synthesis of your microorganisms in their growth medium. And we also discuss the different categories of your microorganisms based on how they obtain carbon and energy. And lastly, we have discussed how the substances can be transported across the cell membrane of your microorganisms. So again, we have here simple diffusion, facilitated diffusion, as well as active transport. So for this video, we will continue our discussion on the different factors which affect microbial growth. So what are the different factors which affect microbial growth? So we have here temperature, pH, oxygen, carbon dioxide, osmotic pressure, as well as light. So let's first begin our discussion on the most significant factor which affects microbial growth. So for any microorganisms, there is a minimum as well as maximum growth temperature. So this can be defined in the range where most of your microorganisms can be able to grow. So typically, the average temperature where most bacteria grow as well as the other microorganisms can grow is at the temperature range of 25 to 30 degrees Celsius. So basically, at ambient temperature, most microorganisms is possible to grow. So you also need to remember that at lower temperature, for example, a chilling temperature which can be achieved at 0 to 5 degrees Celsius, there is slower microbial growth because of your reduced enzymatic activity as well as reduced membrane fluidity. So again, at lower temperature, the different enzymes which break down or catalyzes the bigger molecules, they will have reduced activity at lower temperature. So not only that, the cellular membrane of your microorganisms, which compose mainly of your phospholipids as well as the different fatty acids on its chain, its form will basically be solid at lower temperature. So that is why there is reduced or slower growth rate for your microorganisms at lower temperature. So we have here a figure to you showing the growth rate as well as the temperature of your microorganisms. So the growth rate are the highest or at highest at the optimum growth temperature. Okay, so again, the growth rate, it can be highest at the optimum growth temperature or the peak temperature of your microorganism. So at the optimum growth rate or optimum temperature, there is the highest optimum growth rate for your microorganism. So the lowest temperature at which the organism can survive and replicate is its minimum growth temperature. Again, the lowest temperature at which the organisms can survive and replicate is its minimum growth temperature. In comparison to that, the highest temperature at which growth can occur is its maximum growth temperature. So you need to remember that once the microorganisms have achieved their optimum growth rate, further increase of temperature will basically degrade, okay, will basically degrade the growth rate of your microorganisms, or there will be decline in the growth rate due to thermal denaturation of protein. So when you say thermal denaturation, most of the proteins present in your microorganisms in the form of nucleic acid, they will be denatured or mamatay sila due to exposure of higher temperature. So that basically explain why most of your microorganisms will decline or die at higher temperature. So different microorganisms can be categorized according to the temperature range or temperature requirement at which they grow. So again, we have here another figure showing to you the growth rate versus the temperature of the microorganisms. So first, let's discuss the first classification or category of your microorganisms based on its temperature requirement. So the first one we have here is psychrophile or psychrophilic microorganisms. So psychrophilic microorganisms, these microorganisms are cold loving. So they have optimum temperature of negative 10 to 20 degrees Celsius. So basically, they have low temperature requirement and thus they are cold loving. So again, those cold loving microorganisms which grow below 20 degrees Celsius, we call them psychrophiles. Okay? Moving on, we have another classification of microorganism. This is your mesophile or your mesophilic microorganisms. So, in comparison to your psychrophile, which are cold loving, mesophiles are moderate temperature loving and they have optimum temperature requirement of 25 to 40 degrees Celsius. So most of your microorganisms that can grow at room temperature, basically those microorganisms are what you call mesophile or mesophilic microorganisms. Okay? And moving on, we have thermophile or thermophilic microorganisms. These microorganisms are heat loving, so they can grow best at 50 to 80 degrees Celsius. So that is the temperature range where those microorganisms have optimum growth rate. So that is your thermophiles. And then we also have here your extreme thermophiles or your hyperthermophiles. So these microorganisms are extreme heat loving and they can have optimum growth temperature at greater than 80 degrees Celsius or more. So again, we have here the different classification of your microorganisms based on their required growth temperature. So again, your psychrophile, they are cold loving and they grow best at temperatures less than 20 degrees Celsius. Then we have here your mesophiles or your mesophilic microorganisms which are moderate temperature loving. So they can grow at 25 to 40 degrees Celsius. So basically your ambient temperature. Then we have your thermophilic microorganisms or your thermophiles which are heat loving. So they can grow at 50 to 80 degrees Celsius. And lastly, we have extreme thermophile or what you call your hyperthermophile, which are extreme heat loving microorganisms. So this microorganism can grow at greater than 80 degrees Celsius. So again, those are the different categories of your microorganisms based on their optimum temperature requirement for them to grow. So again, temperature is an important factor which affects the growth rate of your microorganisms. So now let's move on to pH as another factor for the growth of your microorganisms. So we have here another figure to you showing the growth rate of microorganisms at specific pH. So the pH of maximum growth rate is called as their optimal growth pH. So the peak here is their maximum growth rate. Okay, and we call this peak here as the optimal growth pH. So based on this optimal growth pH, we can separate the microbes into three groups. So we have here acidophile, neutrophile, as well as alkalophile. So acidophiles, these microorganisms, as you can see here in this figure, they grow best at temperatures less than, or sorry, your acidophiles, they grow best at pH requirement below 5. So pH below 5 is the optimum pH requirement for your acidophile. Okay? Moving on, we also have here your neutrophile. So neutrophiles, they grow optimally at pH between 5 as well as 9. So the optimum pH requirement for your neutrophile is between pH 5 and 9. Then we also have here your alkalophile. So these microorganisms, they grow best at pH above 9. So 9, 10, 11, 12, 13, 14, those pH requirement, you can observe that your alkalophile will grow best at that temperature. So again, pH is important, another important factor which affects the growth rate of your microorganism. So we have acidophile, which grows best at less than 5 pH, and then you have your neutrophile, which grows between 5 to 9 pH, and then we have here your alkalophile, which grows at greater than pH of 9. Then we have another factor which affects the growth rate of your microorganism. So we have here oxygen. So anaerobe is an organism that grows in the presence of your oxygen. So it uses oxygen as a terminal electron acceptor in the aerobic respiration. So without oxygen, the electron transport chain will not occur because again, oxygen is the last electron acceptor. So those microorganisms that require oxygen, we call those microorganisms as aerobic. Okay? Aerobic microorganisms, they require oxygen. Then we have here microorganisms that grows in the absence of your oxygen. We call those microorganisms as anaerobic microorganism. So again, when there is oxygen, your microorganisms when your microorganism require oxygen, so we call them aerobic. Then if they don't require oxygen, we call them anaerobic. So we also have here your obligate anaerobics or obligate anaerobic microorganisms. So these microorganisms, they cannot tolerate oxygen at all. So hindi ko sila pwede mag-grow nga may ara oxygen. Basically, with the presence of oxygen, either matay sila or they will not grow at all. Okay, so we call those obligate anaerobics. Then we also have here your facultative anaerobes. So They are able to act like aerobes in the presence of oxygen, but have the added facility of being able to survive when conditions become anaerobic. So your facultative anaerobes, they have they can act as anaerobic in the presence of oxygen, so they can grow with the presence of oxygen, but they can also survive when the condition of your growth media uh, is anaerobic. So we call those microorganisms as facultative anaerobes. So again, we have aerobic microorganism, anaerobic microorganisms, and then we have obligate anaerobes, and then lastly we have your facultative anaerobic microorganisms, um, which can grow with the presence as well as with the absence of your oxygen. Then, we also have here another category of microorganism. We have aerotolerant anaerobes as well as microaerophiles. So, aerotolerant anaerobes, these are microorganisms that are basically anaerobic. So, they don't require oxygen to grow, although they are not inhibited by atmospheric oxygen. So they do not utilize it. So again, your aerotolerant anaerobic microorganisms, they are basically anaerobic, and they are not inhibited by your atmospheric oxygen since they don't utilize it. Then we also have here microaerophiles. So these microorganisms, they require oxygen, but they can only require low concentration of your oxygen at 10 to uh, 2 to 10%, finding higher concentration harmful. So your microaerophiles, they only require small amount of oxygen to grow. So higher than 10%, basically it will be harmful for your microaerophilic microorganism. Then another factor we have here in addition to your oxygen is carbon dioxide. So what are the different categories of microorganisms according to their carbon requirement? So we have heterotrophs, autotrophs, and then other nutritional categories. So on the basis of their energy source and the carbon, most microorganisms, they can be divided into two major classes. So we have here your heterotrophs as well as autotrophs. So the classification of most microorganisms based on their carbon dioxide requirement uh, is uh, is as follows. So you have here your autotrophs. So autotrophs, these microorganisms obtain carbon from carbon dioxide and use light energy or inorganic chemical compounds to produce complex organic compounds known as your autotrophs. So these microorganisms, they can synthesize their own food from inorganic compounds. So most bacteria or most microorganisms that use carbon dioxide as their major or even sole source of carbon are what you call your autotrophs. So in comparison to your autotrophs, your heterotrophs, these microorganisms can utilize light energy, but they cannot use carbon dioxide as their sole source of carbon. So that is the comparison for your autotrophic microorganism as well as your autotrophic microorganisms. So another factor which affects the growth of your microorganism is osmotic pressure. So osmosis is the So osmosis is the diffusion of water across a semi-permeable membrane from a less concentrated solution to a more concentrated one. So the movement of your water across a less concentrated solution to a more concentrated one is what we call your osmosis. So the pressure required to make this happen is what you call osmotic pressure. If a cell were placed in a hypertonic solution, one whose solute concentration is higher, osmosis could lead to a loss of water from the cell, or what you call here your plasmolysis. So this is the basis of using high concentrations of salt or other solutes in preserving foods against microbial attack. So in the opposite situation, water would pass from a dilute or hypotonic solution into the cell, causing it to swell and burst. So again, during osmosis, there is a movement of your um, microorganism from a less concentrated solution to a more concentrated one. So if the microorganisms of if microorganisms are placed in a hypertonic solution, so hypertonic solution is whose solute concentration is higher. So osmosis would lead to a loss of water from the cell. So during hypertonic solution, for example, when there is high salt or high sugar, which again your sugar or salt can be the solute. So when it is in the hypertonic solution, osmosis will lead to a loss of water. So this will preserve the foods against microbial attack. So since there is high concentration of your solute, example your sugar or your salt, osmosis could lead to a loss of water. So since there is a loss of water, then basically it will lead to your um, it will lead to preserving your foods against different microbial attack. So osmotic pressure is important because it is required for your osmosis or the diffusion of your water from less concentrated to a more concentrated solution. And the last factor which affects microbial growth is light. So one category of microorganism that is import that requires light is your phototropic organisms. So phototropic microorganisms, they require light in order to carry out photosynthesis. So again, those are the different factors which affects microbial growth. So again, the most significant factor we have is your temperature. Then we also have pH, in where most microorganisms have optimal growth rate, and we also have here your carbon dioxide as well as osmotic pressure and light. So those are the different factors which affects microbial growth.